Hi, welcome to the Lower Brooks River. This is Katmai Conservancy Ranger Naomi, and I'm here with Ranger Michael Saxton.
Hi, everybody.
And we are going to try and find out who's your daddy here on the Brooks River.
I'm not sure where this name came from.
So, Ranger Michael is doing a DNA study called the Katmai Genetics Study, and we're going to talk about that today. This chat is going to be a half an hour. We're going to take your questions after the chat. Put your questions in the comment section in the live chat channel, and the kind moderators at Explore are going to let us have those questions after we're done with the chat. I remind you that we cannot see the chat, we can't see those comments, so we were going to get them through Explore. So I'm very excited about this chat today,
Mike. Me too.
Oh, good. Because there are three questions that I get all the time. One is, where is Otis?
Okay.
The next one is, where is Bead Nose?
Okay.
And the third one is, what's up with Ranger Michael's genetics study?
Okay.
So, what is the Katmai Genetics Study?
Oh, I thought we were starting with where's Otis.
Where is Otis?
I don't know.
Oh, okay.
So what's going on with the genetics study?
What is it?
What is the Katmai Genetics Study? So the Katmai Genetics Study is part of my PhD dissertation. But what we're doing is we're looking at gene flow throughout Katmai National Park and Preserve, trying to figure out what level of connectivity there is throughout the park, and whether or not these bears are breeding across the Aleutian Range and over into the preserve and not, or if they are more isolated into subpopulations here.
Okay, now we're going to speak a little louder because there is a plane taking off, which never happens.
Never happens.
We have no planes that come here. So what's the complete purpose of the study? I know you're trying to figure out who's breeding where and what our population is made up of. What else do you want to know from the study?
I'm going to give it a second here.
You're going to give it a second?
Yeah,
okay.
Since it's fired up fully there. Okay.
Oh, yeah.
Okay, there we go. A little quieter now.
Okay.
Okay, so the full purpose of the study, as I was just explaining, we're trying to look at gene flow, particularly across the Aleutian Range. So we have these really healthy populations of bears over on the coast, out at places like Howlow Bay and Geographic Harbor. And then we have bears in these populations in places like Brooks Camp that are more on the interior Bristol Bay side of the range. And something that's long been a question for the park is, what level of movement is there between the coast and the interior of the park? I'm trying to keep my hands under control here. My normal expressivity might result in me hitting you in the face, so I'm just gonna...
Just don't hit me in the face. You can use your hands. I don't mind that. I'll go like this.
Okay, sounds good. Okay. Sounds good. Anyway, so we're looking at gene flow throughout the park, trying to see what level of connectivity there is. And that's really important as managers, because we have different variable levels of impact in these different areas of the park. Someplace like Brooks Camp, we have a large human presence. We've got infrastructure developed here. We've got a lot of people coming through. Places like Howlow Bay, Geographic Harbor, we don't have the infrastructure, but we do still have a pretty high level of use. But then there are other areas of the park where we don't have a lot of human use. And then there's areas like the preserve, where there is still, sport hunting and subsistence hunting that's occurring up there. So it's really valuable for us to know, are we managing one connected population, or are we managing multiple smaller subpopulations?
So how far is it from here to the coast, to Howlow Bay?
From here to the coast? Oh, that's an interesting question. It's not that, so let's see, the park is about 100 miles across at its widest point. From here to Howlow Bay, boy, maybe, I've never actually looked specifically, maybe 40 or 50 miles probably.
So that's doable for a bear, very easily.
Very easily doable, yeah. Very easily. Certainly something that they could manage in a day. Really, the more likely reason you'd end up with any sort of a split would be due to one of a few different factors. One could be, you've got phenomenal food resources at Howlow Bay. You've got phenomenal food resources here. And through the center, you've got a mountain range that does not have those salmon runs, doesn't have the sedge meadows, can't dig for clams, might get some berries, but when you start getting up more into the alpine zone, you even lose those. There's just not that much food available. So if you've got all these great resources on both sides, it's kind of, well, why move back and forth? And you may have some young males that disperse, or bears in general, they may disperse across that mountain range.
Mm-hmm.
And you also may just have some curious bears. I mean, I think a few years ago, the camera up on Dumpling Mountain, there was a bear that went and sat down in front of it and just kind of sat there staring out and enjoying the view for a while. As we fly around the park, we often see bear trails up in areas where there are not food resources, and you wouldn't necessarily expect a lot of bear activity, but there's very well-developed bear trails in some of those areas. So we may have movement through there, but we're trying to figure out more about it.
So we do have wildlife researchers out there. Have they spotted some of the bears that we recognize here out in Hallow Bay?
So we do not have a presence on the ground that much this year. Some years we've had more of a presence on the ground out there. So Joy was out there for a few years working on her research. I think everybody on the cameras has met Joy before. But the people that have been out there are generally not the people that we have in camp here. I mean, everybody knows Leslie and Tammy that are doing a lot of our IDing. So we don't have the people there that would maybe recognize individual bears. That said, I suspect that most of our bears are not moving back and forth across the coast. I suspect we get some movement, but whether or not it's enough to maintain genetic connectivity is what we're trying to find out.
Ah, okay. So, what are your methods for figuring out about the genetics of these bears?
Sure.
How do you do that?
Sure. Any way we can.
Just like, where does a bear sit? Anywhere it wants to. Yeah. Okay.
Yeah. It's so we start by collecting the genetic material. So as people here are aware, that sometimes that involves the biopsy darting that we're doing here.
Well, you might explain that a little bit more, because not all the cam viewers have been watching.
Sure.
Forever, like me.
Okay.
So what is that?
Sure. Yeah. So we have what we have is a CO2 powered dart rifle, same as what you'd use to tranquilize an animal, but instead of injecting it with any drugs, we use a specific kind of dart that has a larger bore needle. It's about a 2 millimeter bore, and when it hits the animal, it bounces out generally, or if it does stick, it'll work out pretty quickly once they start moving. When it bounces out, it takes a little bit of skin and fat from just that subcutaneous region, and from that you can extract the DNA. So that's primarily what we've been using around Brooks Camp. And then in addition to that, I've been doing hair snares. So throughout the park, when we find rub trees, which is an area where bears go, and you'll find hair from generations of bears rubbing on it.
Right.
So we can wrap barbed wire around those trees. And when the bears go and rub on it, then it'll take little bits of hair out. So then looking at the root of the hair, we can get DNA from that.
Do they change where they rub on the trees when it's got barbed wire on, or they just don't feel
doesn't
it?
Seem to bother them in the slightest. I have watched bears rubbing on these trees, and they don't seem to be aware of it. If anything, it might be a nice extra little scratch. They've got pretty thick skin, so it doesn't seem to bother them.
Yeah, we've seen them getting some big tussles on the cameras, and they don't seem to suffer greatly from that.
Yeah.
So, what are your challenges to dart successfully? I mean, these are bears, wild bears.
Sure. So there's a few different challenges that we're facing. One of them, as I know I've mentioned before on here, is we've had a couple of problems. One is the darts that we were using the first couple of years, we'd have an issue with they would hit the bear, bounce out, and it wouldn't take a sample. Like we know that the dart is penetrating and it's going into the skin, but the way that it was coming out, there was no barb or anything on it inside the needle, so it wasn't holding on to the sample. So it would just hit them, bounce out, and there'd be no sample.
How long did it take you to figure that out?
Well, we knew it was occurring early. We just didn't know what the fix was early on. So thankfully, I was able to talk to a few other researchers around Alaska that have done similar kinds of work, and they were able to point me in the direction of some other resources and other suppliers that had a different style of dart that holds on to that material a little bit better. One other problem we have is, as you mentioned, these are brown bears. They're large animals with the potential to get grumpy and angry when you poke them.
I don't know
So,
why they would.
I can't imagine. So for the most part, when I dart them, the response is huff, maybe trot 10 yards, and then continue grazing. Occasionally, I've had bears that have no response whatsoever. Some of the viewers may be familiar with bear 410.
I was just going to say, I bet 410. 410, for those of you who don't know, was our oldest bear until last year when she stopped coming at age 29. And she is one of those immovable, was one of those immovable objects. I mean, she could, we talked about hazing, she could not be hazed. People would walk by her when she was asleep, and she didn't care, didn't move. So I can imagine. So tell us about trying to dart 410.
So just to put it in perspective, first I'll tell you a story about trying to haze her. Once she was in camp and she was sleeping in camp, and I tried hazing her and I couldn't get her to move, which was not unusual. So I enlisted the help of a bunch of other rangers, and there were 8 of us. We had noisemakers. We all got far closer than I would with most bears, but we're yelling at her, screaming, making noise, banging our noisemakers. She wakes up from her nap. She lifts her head and she looks around at all of us standing there making noise and yelling at her. And she puts her head back down and continues to go to sleep. So you get her to move. So when I darted her, it was on the beach in front of the visitor center. And I dart her, and as I said, most of the time the dart kind of bounces out. Well, with her, it stuck in, and she didn't move. She just sat there. So normally I'm relying on the bear to start moving at that point in order to get it to work out.
Yeah.
She just kept sleeping. So I ended up having to try to, once again, the challenges with hazing, trying to haze her down the beach a little bit to get her to move again so that I can actually get my dart back. It took a little while. It was one of the more challenging dartings that I've had. So that was entertaining.
How about any bears who really didn't like your darting?
So that was the next one I was going to talk about. I have these few that don't care at all, and then I had one. Another bear that many viewers are probably familiar with, bear 856.
I was going to guess that. For those of you who don't know, bear 856, he is the most dominant bear here. And it's not about just his size, because he's not the biggest bear. He owns this place.
Yeah. He was pursuing another male who was pursuing a female at the time, and 856 has been on the we need to try and get a sample from him because he's the most dominant. We expect he might be heavily involved in the genetics of the Brooks region.
Yes, answering that question, who's your daddy?
So really want to get a sample from him. So I see I have this one opportunity, and he's kind of lazily pursuing these other 2 bears. He went under the platform out at the falls, or under the boardwalk out there, and after he passed under, I darted him, and that was the only time that I'm quite convinced if I was on the ground with that bear, he would have killed me. He turned and roared, and sat there kind of looking around like, who the heck dares do this to me? I'm in charge out here. And he looked around for a minute, and he didn't see anybody. So then at that point, he turned and continued to pursue the other 2 bears.
So you do have a sample from 856?
No.
No.
Because that's one of the ones where it hit him, bounced out, and did not take a sample.
Oh, that hurts. So I assume that you really would like to get a sample from 856.
856 is a high priority, but he's kind of challenging. He doesn't often show up underneath the walkways. He usually hangs out more in the river.
He does like to come from the side of the platform. You might try that.
From the side upriver or downriver?
Upriver.
Seen him? Okay.
He's been doing that a lot this year.
It's the challenge there in July in particular is if I dart him upriver the platform, he has to be close enough that I can reach it with my little special grabber tool with a grabber stick duct taped onto it. And if I can't reach it with that, then if the dart falls out, it could be in that closed area. And I don't like to enter the closed area in order to try and retrieve these darts, so I try not to dart bears when they're in that area. I mean, laws and bears and all those strange
staying
things.
alive.
Yeah, exactly.
Yeah, little things like that.
Yeah, minor issues.
Okay. So who have you not darted that you'd besides 856 that you'd really like to get samples from?
He's probably the highest priority target. Some viewers who have been watching for a few years have probably seen me dart 83 on multiple occasions and still not have a sample from him.
Oh, really?
So he is a priority, especially now that I've switched to these new darts. I have had a much better success rate getting samples, so I'd like to get him. 747, I don't have a sample from.
Oh.
Yeah, a lot of the males, they're tougher because they tend to stick to the river more, and we need them to go underneath the platforms because I don't like darting when I'm on the ground. Just in case I have one of those 856 responses.
Silly you.
I know, one of those whole staying alive things again.
Yeah, I know. So, what about darting and getting samples from the supposed children of these big bears?
Well, and that's where we are now. So last year we started to dart more of the subadults and kind of the younger ones, so that we can fill out that that pedigree a little bit and try and get a better idea of all the bears that are in the area. I don't like darting bears when they're too small because it is not a small needle, so I don't want to hit a bear who's really small and have it penetrate further than I'm comfortable with. So I'm usually waiting for them to get a little bit bigger, but last year we did start to get some of the 800s that were getting a little bit bigger. So that's kind of where we are now. We've got a good portion of those older bears. That's still the highest priority, is to fill in those, because those are more likely to be the ones that don't show up again in future years.
Right.
The younger bears, we've got a better chance of getting them in the future. So I'm less concerned with them. They're a lower priority. But we are starting to fill in the tree with those younger bears.
So we and the cam viewers have not seen you darting bears yet. Are you going to be darting?
I've been sneakily hiding. I've gotten 127 samples this year. I'm lying. I have not done any darting yet this year.
Okay.
I am going to be darting, but in the past what I've found is that I have the most success late in the season. After camp closes, there's a few reasons for that. One of them is the bears themselves. The fat layer is a lot thicker. It's a lot easier for me to dart them at that time of year. But part of it is also human activity in the area. I don't like darting when I don't know where all the people are in the area, because you don't know exactly what the response from the bear is going to be.
Right.
And I don't want to upset a bear and then have it go run into somebody in the river or anything like that.
Not a good idea.
Not ideal. So that's something that I'm very, very aware of when I'm darting. And late season, we have a lot fewer people out here, so it's a lot easier for me to dart at that time of year. This year is actually going to be really interesting. We have this fancy new bridge out here, which opens up a lot of darting opportunities that I would not have had previously.
Oh, that's right. I hadn't thought about that as an advantage for the bridge.
Yeah. So, I don't want to dart over the river, because I also like to be able to retrieve these darts afterwards. Right. And if they go in the water, then it gets a little tougher to find them again. But now areas over the marsh marsh is dry this year, for better or for worse.
Yeah.
So all of those areas over the marsh, areas over the closed trail, we've just got a lot more areas that I have to dart from. So I'm hopeful that will allow me to get a few bears that in the past have been tough to get, because they don't come to this side of the river as much.
So bear cam fans, you're going to have a nice fall this year. You're going to be able to watch Ranger Michael darting bears again. I know I enjoyed that a lot. It was fun to see. Although I think maybe darting Otis was a really good entertainment for us.
That was, I was just really happy to finally get one from him. I know that was a lot of people's highest priority, was that I get one from Bear 480, and thankfully last year I was able to get him. So that was fortunate. I remember watching him coming down the river and just being like, oh my goodness, he might actually show up over here. He never comes over here. Maybe I'll get a chance. So that was quite exciting.
That's good. That'll be very important for everyone concerned. So what happens with the material after you collect it?
Yeah.
What's the next stage in the study?
So some of it, the biopsy darts I take back down to where I'm working at Washington State, and I've been doing all the DNA extractions on those samples myself down there. The hair samples have been going to a lab in Anchorage, a USGS lab, who's been doing a lot of the genotyping as well. So after I extract the DNA, I'm sending the material to this USGS lab in Anchorage, and they've been processing the samples and doing the genotyping for me. And that's been going all through the winter and then into this summer.
Okay.
Yeah, and then so we also had some blood samples from Joy's project, and I did the DNA extractions on those as well and sent those off.
Yeah, so that makes me think, like, what is going on with the population in Hallow Bay? Because how do you know whether there's a connection between that population unless you're getting samples out there?
Yeah. So that was those blood samples that we got from Joy's project were from all along the coast. So we're using those as some of our coastal samples. And then we had some hair snares that were set up out on the coast. I did a little bit of biopsy darting over in Amelik Bay, Geographic Harbor area. And then there's actually some old studies that have samples as well that we're hoping to use some of their data as well to help improve the number of samples that we've got from some of those areas.
Okay, great. So are there more high-tech techniques either for gathering material or analyzing that you don't have available to you that would be on your Christmas list?
I guess so the way we designed the study as it is right now, we're using microsatellites, which is a genetics technology that's been around for a little while, and it's still very effective, but you're looking at small sections of the genome. And part of the reason we designed it that way is that there was that 2006, 2007 genetics project out here that used microsatellites as well. And so this allows us to compare to those older samples. Moving forward, in a dream world, it is a bit more expensive, but in a dream world, I'd probably switch to doing whole genome work. I think that likely moving forward, whole genomes, it's getting so cheap at this point that that's probably going to be the direction most wildlife work is going.
Can you clarify that? What is whole genome work?
Sure. So rather than, like I said, with the microsatellites, you're looking at some small segments, usually around 16 to 21 segments of the genome. These little repeating sections. With whole genome, you're looking at the entire genome. So it's a huge data issue. I mean, you have to store, so it requires a lot of storage and a lot of processing capacity in order to work with it. But that's what I've been doing more so in grad school. I've been looking at the transcriptome, which we don't need to go into that.
Okay.
But
I have
anyways,
no idea what that is.
Yeah. We're looking at using these much larger data sets. They offer a lot more information, and I think moving forward, if we can go into using whole genome stuff here, it will allow us we'll have a lot more power to work with, and there will be a lot more questions that we can answer if we're doing whole genome.
Okay. I had a question about getting samples from known siblings, right?
Okay.
Over the years, when you get, there are a number of bears here that we know are from the same litters. Have you consciously tried to sample from those? I
have not made any exact effort to sample them, but I am trying to get as many bears as I can out, here at the Brooks River. So hopefully at some point we're able to fill in, get samples from most of the bears out here, in which case, yeah, we'd have samples from a lot of siblings. As it stands right now, though, no, I have not made a specific effort to target siblings. Yeah, because a question that would occur often is, well, the possibility exists that they don't have the same father. So if you have the siblings, we could then figure out who's your daddy. Yeah, and I would fully expect that many of the siblings don't have the same father, because they do have multiple paternities pretty regularly. So will your study be insightful about inbreeding and potentially down the line any effects of inbreeding? So we are going to look at inbreeding coefficient when we're analyzing this. This is sort of a pet project of mine. I'm curious, and this will be basically preliminary data to look at inbreeding in different locations throughout the park. I'm curious if we see differences in the level of inbreeding in some place like Brooks versus Hallow Bay versus someplace where we don't have a lot of human presence. And if we do find that there is a lot of inbreeding in some of the areas with high human use, then that would suggest that we need to look into why do we have this issue. Just that there is more inbreeding location versus another, that doesn't necessarily give us a reason why. And if we do find that Brooks has high levels of inbreeding, there could be a lot of different reasons. From either it could be that there's so many people around here and there's only some bears that are willing to tolerate it, or it could just be the fact that this salmon run starts so early it overlaps with the breeding season, and bears we know have pretty strong site fidelity to feeding locations. So if the same bears are returning year after year to that feeding location, then they may just be breeding with each other just because they're returning for the food source. It may not have anything to do with people.
Now, I have a question which it has to do with recognition of relatives by bears. I mean, I know there are studies of dogs who have very good sense of smell, that they can smell down to a chromosome and potentially recognize relatives. Have there been any?
I'm unaware of any studies like that existing. I wouldn't be surprised if they can recognize each other in some way, shape, or form. But I'm not aware of any studies that have been done on it. Okay,
I think that's something for you to do in the future.
Oh, there we go.
There are lots of work.
I don't have enough to do yet, so.
No, I know. We'll get you a third job here.
Perfect.
So, what's the future of your study? I'm going to ask you the question, do you have any results to share with us?
Okay. Well, let's start with that before people get too on the edge of their seats. No, I don't have results to share with you yet. These sorts of things do take a long time, and I got an email recently from the lab that is processing the samples, and things are going well. I was a little bit concerned with some of our hair samples, that they weren't going to be able to extract DNA very. It seems to be running pretty smoothly. You only expect 60 to 70% of your hair samples to actually be able to extract DNA. So that's, but it seems like it's all going reasonably well. Things over the winter did definitely slow it down a little bit. I mean, it is a USGS lab, so the government shut down over the winter, and then also there was the earthquake in Anchorage, and those things definitely slowed it up a little bit. But it is going very well. It sounds like hopefully I'll have data back in the next month or two, and at that point I can start doing analysis. So hopefully over the winter or by next spring, maybe I'll actually have a little bit of this information. I know people have been patiently waiting for years now, and we're excited to, trust me, I'm as excited as any to finally be able to do some analysis on this.
Okay.
And there was a first part to that question. Oh, future of this study.
Future of the study.
So the portion that I was doing for my dissertation, I'm done with at this point. However, as I mentioned, I am still planning on doing some sampling this fall. My vision for this project going forward is that we turn this into a long-term population health monitoring, and not just here at Brooks, but throughout the park. Try and do sampling every 5 years or so, and be able to monitor the genetic health of the population moving forward. There's a researcher in Canada, Gordon Stenhouse, who has some really, really incredible research that he's been able to do, where he started similar to we did with the biopsy darting and stuff like that. And then moving forward, he actually went to, once he had the basic framework of the pedigree put together, they started doing it as a citizen science project and collecting scat samples. And so like every year they send off maybe 10 scat samples to just help keep filling in the population information out there.
Something for you to keep in mind, you people who are going to come visit Brooks.
Come and collect scat.
Yay! So great. So one more question before we get to our viewers' questions. Are you known by the bears as Mr. Dartman, and do they hate and fear you?
No, I wear a Tammy Carmack mask every time I'm darting. No, I suspect that the bears might know me. However, I also suspect that if they have any association with me, it's far less to do with the darting and far more to do with the 7 years of chasing them out of camp that I've been involved with. So I would not be surprised if they know who I am. I've certainly dealt with bears. I can think of one in particular, a black bear in Sequoia Kings Canyon, that she definitely knew the rangers. Anybody else could walk up and she'd sit there and continue doing exactly what she was doing. And as soon as a ranger walked up, she was gone. So she seemed to be aware, and I wouldn't be surprised if the bears here also are pretty aware of who we are.
They have to go out of uniform for that, right?
Exactly.
To get out of the green and the gray.
I'm sure they know my smell at this point, but...
So I'm going to take the phone from our camera operator, my guest, my friend from college, Barbara, who's helping us out, and see your questions. Oh, we got a lot of questions, Michael. So let's see if we can stump the ranger.
Oh, that's very. They just started off. The answer is I don't know. Whatever the question is, the answer is I don't know.
So helpful. You're so helpful. So the number one question, which I told you would be the number one question, does 480 Otis have any offspring?
I'm guessing yes. As I said, we don't have the results in yet, so we don't know. Don't know at this time, and I can't imagine he doesn't. But we'd also have to have samples of those bears. So we'll see once the analysis gets going this winter.
And here's another one. Michael has an additional responsibility today. Radio is on.
Oh yes, I apologize for that, everybody.
So he's the man in charge today. So we go on. So this is sort of the second most popular question, and it came in number 2. Do you know who 503's father is? We have some guesses.
I have a suspicion, yeah, but once again, without the results back, no, I don't know. Don't know who his father is yet. And if my suspicion is right, I do not yet have sample from his father, so we'll see.
That's your assignment. Should you choose to accept it, is to get that sample.
I would love to. As I mentioned, he's sort of priority number 1. I'm guessing it's 856. I don't know that by any means, but yeah, that's my guess.
A few similar physical and behavioral characteristics there.
Yeah.
Okay. Oh, this is good. Have you found out anything in your studies that you didn't anticipate at all? Maybe in technique or...
I mean, methods is always a learning process. I mean, heck, the first time around wouldn't work. That was a learning experience.
Right.
Things that surprised me, I guess the response of the bears to darting surprised me a little bit. It's less of a response than I anticipated it would be. As I mentioned, for most bears, it's kind of a puff and trot off 10 yards or so. And boy, that's the level of response to expect. But I would have a much stronger response than that. So far I've avoided it. I'm waiting for the bears to figure out how to operate the dart gun, then I'm going to be in trouble. But yeah.
Okay. This is another good question. How long is the dart material you get viable?
Okay, very good question. So when we use the biopsy darts and get that skin and fat sample, I actually take it and put it into a solution called RNA later. And that's actually designed to preserve nucleic acid. It's stable for quite a while. Freezer, and when you get it into a -20 or a -80 degree freezer, you've got a long time before you need to worry about it going bad. If you get it into -80, I could probably analyze it 20 years from now and still have viable material.
We don't want you to analyze it in 20 years.
I'd rather not to.
Yeah,
I'd rather not.
Okay.
Yeah.
Okay.
The hair samples, sorry, I'm going to jump in. The hair samples are a little bit different. With hair samples, and this is part of why you don't have the same extraction rate with hair as you do with tissue. And besides that, there's not as much DNA to begin with. It's they're going in with like a desiccant or something, and then you want to try and get them into a freezer within a week or two. Otherwise, it can be tough. The biggest things that degrade DNA is either water or UV. So when you've got a nice wet landscape out here, and then it gets sunny every once in a while, like right now, it does tend to degrade the DNA as it's sitting out on the trees. So that can be a little bit tougher.
So is that a challenge this year because we've had so little rain and it's sunny?
Well, because I don't have hair snares out this year.
Okay, I got that answer. Right. You gonna have other rangers helping you out with the darting this year?
I will likely have a few people from the bear management team around helping me just with spotting and everything like that. Tammy will likely be helping me with IDing the bears because I'm terrible at IDing bears. People can ask me what bear it is all day long, and unless it's one of the like 10 or so that I know, my response is going to be that I'm fairly confident it's a brown bear. Fairly confident.
That's his standard answer. I ask Michael all the time, and I get it's a brown bear. Is there a site where we can go and see the bears that have been successfully darted and any of the results of your studies so far? You published anything?
Not that I'm aware of. There's not a site that exists on. I hear there's like a wiki page for the webcams though, so maybe on there might be a list of what bears have been darted. So I wouldn't be surprised.
Check on the chat with your fellow webcam viewers.
As results become available, then we will happily put out any parentage information as soon as we get that information. So we'll put it out through Explore, through Facebook. Basically, I'll let our interpretation team have they choose to.
I can't wait personally. Let's see. Do you know, since you're not great at bear IDs, do you know which of the 800s you've gotten so far?
Ooh, I don't remember off the top of my head. I actually think I got a couple of 900s last year. Who did I get? I want to say 910.
Mm-hmm.
And maybe another one that was either 902 or 903, but we weren't sure at the time. But happily, one of them's female, so once we get the genetic information back, it'll be pretty easy to tell which one it was.
And for those of you who don't know, those are offspring of a 128 and 409.
Yeah.
So those are great samples for you to have.
But yeah, the 800s, I don't remember off the top of my head who we've gotten. Yeah.
So I'm trying to see if there are any things that we haven't covered. Well, someone wants to know exactly where Margot Creek is.
Margot Creek is...
Because a lot of bears are there right now. And the reason for that is dry summer, and there are the creeks and small are very dry and the water's low, so it is really easy pickings for salmon fishing there for the bears. So why walk 12 miles when you can just stay where you are and fish? So where exactly is Margot?
So Margot Creek is about, so I'm just going to point, it's that direction. So it's about 12 miles down to the east, 12, 13 miles down the shore to the east. River, Iliac Arm.
So it's towards the coast, right?
It is towards the coast, yes.
Okay, so might you get a mix of bears from there and a genetic mix? Maybe you can do some darting out there.
So we actually had hair snares out there for 3 years. So we have some samples that we collected over there. It is closer to the coast, closer to Savanowski Pass over there. So yeah, it's that direction.
Okay. I think that's about, here's another one that I like. Have you managed to, we know you've managed to dart 480. Have you darted 755, and will we ever learn if they are related?
755 I have not managed to get yet. He is on my wish list. I don't think he hasn't been monitoring the last few falls down the lower river as much, at least. Maybe he's been hanging out at the falls.
He's been at the falls until, for those of you who don't know, this is, 755 is otherwise known as Scaredy Bear, who hangs out in the far pool. He got his name because he's a Scaredy Bear. And he looks, in a lot of people's opinion, a lot like 480 Otis. I mean, he has the patch on the side. He's even got a ear now, similar coloring. So by observation, we wonder, is he related? So that'll be the next question you'll get all the
he's
time.
on the wish list, but he is, as you mentioned, he tends to hang out on the far side of the river, not at places that are easy for me to dart.
Yeah.
So unless he shows up, side of the river. So if he shows up in the community, especially with this new bridge, but as he hasn't down in the lower river much in the fall, so we'll see. Be nice.
Well, your questions that we received, we got some more on the chat. I really want to thank Ranger Michael for joining us today. It was so interesting, and coming back by popular demand to explore. I am Katmai Conservancy Ranger Naomi, and this is Ranger Michael, and we're going to say thank you very much for joining us today here at Brooks River.
Thanks, everybody.
Don't turn it off.